All right. To me, some of the most exciting advances in DNA technology have to do with what I call DNA profiling. And this is finding and analyzing DNA. And so we'll go through a couple techniques here and I'll explain some of the applications. So remember, all cells use DNA. It's a double stranded molecule that's built from nucleotides that contain adenine, thymine, cytosine, and guanine. That these nucleotides form specific base pairs, and if you know one side of the strand, you know the other side. All cells use the same mechanism to copy DNA. They use DNA polymerase, they separate the two DNA strands, and then they use each as a template. And using complementary strands, they build new double stranded molecules. We can do that in a test tube, and that's the basis of DNA profiling. So, first, let me say you can find DNA. Okay, DNA in a mixture of other types of molecules has certain chemical components that you can find. You can put probes in there that will bind to DNA. And so you can pull DNA out of a complex mixture of stuff. Okay, some probes can be very, very specific. Others will bind just any DNA. And so it's easy to get DNA, to separate it, and to purify it. So you can, even if there's just a little bit of DNA in a mixture, you can find it. And then, using something called the polymerase chain reaction, you can copy DNA. You use an enzyme called DNA polymerase. It's a heat-stable enzyme from a bacteria called Thermus aquaticus. It's a hot spring bacteria. That's a picture of it there, and there's the hot spring. Then you use primers and nucleotides, and you take your source DNA and you put it in this thermal cycler. And this thermal cycler heats it till the strands separate, and then it cools it down, and the enzyme works to copy the separated strands. And then you run through these thermal cycles, and you can make unlimited copies. And so starting from just one DNA molecule, you can make, in a couple hours, you can make trillions of DNA molecules that have the same identical sequence. Okay, you can sequence DNA as well. Okay, and again, using the same enzyme, DNA polymerase. Okay, you take your source DNA, you add the regular nucleotides, and then what are called chain terminators. These are special nucleotides that are similar to adenine, thymine, cytosine, and guanine, but they have a fluorescent dye labeled to them. And when you put a chain terminator on, when the enzyme DNA polymerase adds a chain terminator, that terminates the copying, and so it makes shorter copies of a DNA molecule. So these short fragments then that you've made can be separated by size with something called electrophoresis, and a computer analyzes the fragments. Okay, there's the machine there. This is animated. And so again, you use this sequence and you separate these labeled things in a gel. And so the shortest things will migrate fastest. And you know how they end because they have these radioactive dyes on them. And so you can just read the sequence by reading which type of dye is there first. So you can find out the sequence of unknown DNA. Okay, so these are really powerful techniques. And together with a couple other kind of DNA methods, you can identify DNA. And in particular, we can use probes for regions called short tandem repeats when we're trying to identify individual people. We could also use what's called restriction fragment length polymorphisms. These are just looking at variable regions of DNA, and you can identify individuals really exactly. And you can do this from incredibly small biological samples, from blood, hair, saliva, semen, even individual skin cells. And so these methods are really, really powerful and really, really exact. They give you, you can distinguish between family members. You can distinguish between thousands of individuals in a single project, and So this has really been a big advance for our criminal justice system because we used to rely on things like footprints and bite marks and blood types. I mean, in fact, some of the best evidence was blood type evidence because you could get 8 specific blood types by looking at blood samples, and that was considered the best scientific evidence. But even the rarest blood type has millions of people that have it, so it was really poor. And so once we started using DNA profiling on biological samples involved in crime scenes, we started realizing that lots of times the samples that we thought incriminated a person actually said it's not them. And so in Illinois, when these techniques first came online, we recognized that we were overturning more convictions than we were executing people for capital punishment. Um, and so Illinois suspended all capital punishment because we realized that despite what juries said, despite what we thought the evidence said, we were often quite wrong. And uh, in cases where there is biological samples, we can tell if those samples come from the person that's convicted. And in fact, the Innocence Project has been using DNA samples to look at convictions, and they've overturned 343 convictions. And even more importantly, the samples have often led to new convictions. That because we have the samples of convicted criminals on file, we can often link that, and that's much better evidence. And what we found is often that small numbers of people are responsible for lots of crime. And so this 147 new convictions is actually for many new crimes, and so it's really been a plus. And I'll have a little link here that'll give you some examples of this. Okay, so in summary, we can make unlimited copies of any DNA sample. We can find specific DNA sequences to analyze, and we can determine the base sequence of any DNA. Okay, this has been really great for criminal justice system, gives us much better, much more reliable identification of suspects and linking samples to crimes. It helps us identify samples of unknown material, unknown plant, animal, human material. It helps us find new genes to analyze, and it helps us reconstruct the past. We can look at the DNA of extinct species from bones and even in fossils. So really big advances. Okay,